em inglês. Passo, então, a palavra para você.
Obrigado, Maria Magdalena, or gracias. Thank you. Thanks also to Igor and Leandro. Yes, my Spanish is not so good, my English is bad, and my Portuguese is probably really, really bad. So I will try to speak in English. But I don't know how you manage this. If you want to interrupt or something, just give a voice up there, because I'm not going to see you from during the presentation because of the screen share. So, or
No,
I can just
we will
speak
leave
at
all
the
the
end.
questions, all the
Perfect.
questions for the
For
final.
the end.
Okay,
Perfect.
great.
So thanks for the introduction. Then I will start with the presentation. As Maria Magdalena was saying, I will speak or talk about alternative splicing regulation in plants, the mechanisms and the consequences of alternative splicing. So first, yes, I'm not a professor. Thanks for the invitation anyway, but I'm a doctor in molecular biology from the University of Buenos Aires. And even though I'm giving some classes, I'm not having a course on my charge here in Argentina. So let's start with this, what splicing means. Then we can go to alternative splicing. And as you may already know, the RNA polymerase II is transcribing the DNA into an RNA molecule. We can see, or we can depict this RNA molecule as a precursor messenger RNA molecule that has a particular sequence. And in this sequence, the cellular machinery will recognize different parts. The boxes are called exons, and the lines are named introns, and the spliceosome, the cellular machinery that recognizes the introns and eliminates or excises them out, is joining the exons together, and it's giving the messenger RNA, the mature messenger RNA, as a result. So alternative splicing is actually having options for the splicing process. The options would be, in this case, for example, that in a particular cellular context, we can recognize these two introns as introns. Then these are eliminated, the exons are joined together, and we have this isoform 1 with the 3 exons together. But in some other cases, you can have, or the cellular machinery would recognize the whole middle thing here as a big intron. It will eliminate that as an intron, giving place to the isoform 2 without this purple exon here. So this is what we call an exon cassette. It's an alternative exon, and this would be exon skipping. This would be exon inclusion. And as you can see, we can have from one particular precursor mRNA, from one particular gene, two different isoforms. So alternative splicing is actually a way to increase the transcriptome diversity and would be a way to increase the proteome diversity. In this case, we have a kind of a human, let's say, and on the other case, we can have a corn. So proteins or cellular products with different function. Now, that's one flavor of alternative splicing, the exon cassette or the exon skipping. We can also have alternative 5' splice sites. In some conditions, the cell will choose this 5' splice site. In other conditions, this one. So you will have two different options of the mRNA of this gene. In this case, we have alternative 3' splice sites. And there is another case. It's not very common in animals in general, but it is very common in plants. That is intron retention. It's different to the others in the case that here there is no a competition between different splice sites. What you have here is either the recognition of the intron and the elimination of the intron, or in the other case, the intron is not recognized as an intron, and then it's not eliminated and it's retained in the transcript. So these are the main forms of alternative splicing. There are more. You can have mutually exclusive exon cassettes, or you can have excitrons that are like introns, but in the excitrons are mainly coding. But how this is regulated in the cell? As I was saying, the spliceosome is the machinery that catalyzes the intron removal, the recognition, and the removal of the introns. And in the RNA, you have sequences that can be recognized, so factors in cis that can be recognized to use one particular splice site or another. You can also have factors in trans, like the components of the spliceosome, but also SR proteins. These are serine arginine-rich proteins, or heteronuclear ribonucleoproteins that can help in the recognition. They bind to different sites, like splicing enhancers or splicing silencers in the exons and in the introns, and they can help the spliceosome machinery to recognize different splice sites. And also, you can have, this is kind of similar to this, a secondary structure that is helping or not helping at all to the recognition of some particular splicing sites. The classic view, or the way we can call this post-transcriptional alternative splicing regulation, is that, as I was saying before, that you have the RNA polymerase II that is transcribing the whole region of a gene. Then you have the precursor RNA, the pre-mRNA, and after the transcription, you have capping, splicing, and polyadenylation. These are all the processing steps. Capping is the addition of the modified guanosine in the 5' end. Polyadenylation is the addition of the A to the end tail in the 3' end of the mRNA, and splicing will be the elimination of the introns to give place to the mature transcript, the messenger RNA, that is then exported to the cytosol for translation. This is actually true for mainly in vitro situations, and it happens, but with low efficiency. What happens in vivo, in the real scenario in the cell, is that the DNA is transcribed, but while the RNA is being transcribed, the regions are being recognized. So the capping, the splicing, and the 3' processing are happening while the transcription is also happening. So these are co-transcriptional processes. This is the scenario in vivo, and it happens with high efficiency. This is also leading to another way to regulate alternative splicing, that would be the possibility to affect transcription and in turn affect splicing. For example, in this case, we have fast elongation. You have a weak splice site, strong splice site, and these are transcribed almost at the same time. So this is mate. I cannot give you through the screen, but we are big fans of mate in Argentina. Then you have a competition between the weak splice site and the strong splice site. Weak or strong are that are recognized in a very frequent way or in low frequency. And if you have this competition, then the strong side will for sure win. And then what you end up is with a transcript that is not including this exon. So you eliminate the whole region here as an intron. But if you have slow elongation rate, then you will have time, the spliceosome will have time to recognize this weak 3' splice site, eliminate that intron first, and then to recognize the strong splice site in turn to give place to the inclusion of this exon. So we have the sequence, the polymerase elongation rate, and the secondary structure, and the factors in trans, all of these factors that can alter, that can modify, modulate the alternative splicing pattern of a gene. Just for you to have in mind, this change in the polymerase elongation rate could be related to the chromatin marks or to, for example, the phosphorylation state of the polymerase. And what are the outcomes of alternative splicing? So as I was saying before, we have all these flavors like exon skipping, intron retention, and so on. And all these flavors will give you different fates, different destinies for these molecules. For example, if in plants mainly, if you have intron retention with stop codons in the intron, then you mainly have a transcript that will be retained in the nucleus. If it's not retained and if it's going out, then that transcript will be recognized by the nonsense mediated mRNA decay machinery and will be degraded, eliminated. So the decision of alternative splicing, the outcome of alternative splicing will determine the fate of the RNA molecule, whether it's exported or not, whether it's translated or not, and whether it's degraded or stabilized. And what about the frequency of alternative splicing? When I started my PhD a year before starting it, we knew, or we thought, that in Arabidopsis we had more or less 7% of transcripts with alternative splicing. This is Spanish, sorry. While in humans, it was reported that more or less 30% of the transcripts or the genes were having alternative splicing. Then after a while, some years, 3 years actually, in Arabidopsis, it was discovered that more or less 20% of the transcripts were having alternative splicing, while the frequency in humans was also going up a bit to 60%. And then some years later, and this is more or less the number we use now in the papers, we say that more than 90 or 96% of the genes on humans are alternatively spliced. And in the case of plants, then it's about 60% of the genes with introns. Anyway, this is probably a subestimation or underestimation of the amount of alternative splicing in the Arabidopsis genome, because it's only taking into account some particular conditions in normal plants, wild-type plants, and it's not taking, as it's happening in humans, single cell sequencing and all the sequencing that is done in humans. So this number would probably grow up a bit more in the next years. And of course, this is not because the alternative splicing was increasing in Arabidopsis during these years, but because the technologies and the methodologies available to analyze and recognize alternative splicing events was growing a lot in the field. So alternative splicing is a rule in animals. We knew that before, and now we know that it's also a rule in plants. It's not just an exception. And then what about its conservation? Then you can see here the 5' splice site consensus. It's a logo. And then the 3' splice site consensus sequence in Arabidopsis and in humans. And it's, as you can see, they are quite similar. So the sequences in Cs at 5', 3', and branching points are conserved between animals, plants, and in all the life kingdoms where splicing is there. Part of the machinery is also conserved. So we have the same spliceosomes in Arabidopsis and humans, and similar factors like SR proteins and HNR and Ps. But there are some clear differences. For example, the introns in Arabidopsis are much smaller than those in humans. In humans, an intron of 10 kb is quite common, and an intron of hundreds of nucleotides is very uncommon, very unlikely. And in Arabidopsis, a normal intron would have around 150 nucleotides. So they are very short. And why plants were? Just going from molecular biology to physiology and development. In plants, we have these beautiful light responses. So we use Arabidopsis thaliana, these model organisms that's growing everywhere except in the culture chamber that we use, that the conditions are not so nice for them. But here you can see it in a row, and it's quite okay. And these are examples of how this plant is growing in, for example, light and complete darkness after germination. So here you have photomorphogenesis, and here you have scotomorphogenesis. Then these are plants that were under a shadow, so under other plants, and here you can see how the petiole is growing and it's being extended and enlarged to trying to collect more light. And then here you can see plants that were exposed to long day conditions or to short day conditions. And this is the flower of Arabidopsis. So in long day conditions, it's flowering quite early, and in short day conditions, it takes much more time to flower. So light, it's not only the source of energy for plants and the source of carbon compounds, but it's also a cue, an environmental cue that is adapting and modulating the development and the morphology of the plant to the particular environment. And as you may know, life on Earth, well, if you don't think that Earth is flat, then this is what we think Earth looks like. And you have 24-hour days, then you have light and dark periods. In the summer, you have more light. In the winter, you have less light. And this is not new. So it's happening for about 4 billion years, when the Earth was actually generated after a collision between two planets that gave place to the moon. Since then, more or less, we have seasons, and we have these 24-hour days. They were a bit longer before, and now it's 24 hours. And after a while, it's like 3.5 billion years ago, since then we have life. And the first kind of life that we have here in the Earth was prokaryotes, cells without nuclei. And a few billion years after that, photosynthesis appeared. And with this, atmospheric oxygen appeared. And this is something huge for the planet. So before this, the planet was even looking with a different color, because there was no iron oxidized, for example, in the atmosphere. And oxygen was not nice for a lot of organisms that were living there. So as soon as the oxygen was rising, some of these organisms were dying. But at the same time, oxygen led to ozone that protected life from UV irradiation. And because of that, there is land life today. So after this, we have the eukaryotes, and some years ago, biology, and then the papers. So what I was saying about oxygen, which is kind of anecdotic, is that the prokaryotes that were creating this oxygen did that in waves. So it was going up quite fast and then going down because they were dying because of the oxygen. And that's related to photosynthesis, of course. Then photosynthesis in some extent allowed the presence or the beginning of eukaryotes on Earth, since photosynthesis and oxygen allowed to use to generate more energy, and also multicellularity. And at this point, we got this heterosis in the cyanobacteria that allowed to put apart the oxygen from the places where the nitrogen was being fixated. So this was a huge advance in evolution, and it was more or less followed by the apparition of land plants, and because of that, the oxygen levels are today what they are. So because of this, we have plants inland and we have animals, and because of this, there is discourse and there is us on Earth. And from eukaryotes, what it's important to consider is that symbiosis is the most relevant thing that happened into the lives of the eukaryotes. So we have the nucleus that might have come from a symbiotic event or not. That's not very clear, but there is some bibliography saying that alpha protobacterium were generating this nucleus or something like that was being used there. And there is something interesting that we have this. Sorry, the alpha protobacterium was related more to the mitochondria, as you can see here, and something related to anarchia was actually given the nucleus. But what's interesting in the plants, or in the plant part of the life, is that there was another symbiosis that was related to a cyanobacterium that gave place to the chloroplast. So that's why plants are doing photosynthesis. And symbiosis, I don't know if you are familiar with this paper, but someone actually, these authors here injected microalgae, and also I think they also tried with chlamydomonas and into a zebrafish, and they illuminated the brain of the zebrafish in a condition without oxygen, and because of the algae and the photosynthesis that was going on there, those fishes were kind of recovered after an hypoxia condition or an hypoxia event. So symbiosis is just a condition in which two organisms kind of live together, one inside another, and in this case, and that's beneficial for both organisms. Since evolution was using symbiosis in eukaryotes since the very beginning, there are for sure some communication between these different organisms that are relevant for the perception of the environment. So anyway, to continue with the light and the light perception, which is important in plants as is the main regulator of the development and morphology of the plant, we know that this environmental cue, light, we can detect the presence or the absence of this cue, the intensity, if you have a strong light or a low light intensity, the direction from where the source of light is coming, the duration of the event of illumination, and the wavelength. It's related to the color of the light. And another thing that is important to consider when we talk about plants is that there is no possibility for them to run away from something that is not nice. Like in the case there is a source of light that is too intense, they cannot go away. So they have to adapt and they have to manage in order to survive in that particular place. So here you can see the development of this plant with this source of light in the upper part of the video. This is Arabidopsis actually, and this is the flowering of Arabidopsis, and you can see the movement of the leaves and everything. Then light is actually regulating gene expression, and that's regulating the development of the plant, as you can see throughout the video. It's regulating the flowering, it's regulating the form, the morphology of the plant and how the leaves are growing and moving during the day. So because of that, and because we knew that plants can sense the direction of the light, also the presence or absence of light, the intensity of the light, this would be 0 anyway, but in this case there is a shadow, so the light is less intense, but also the wavelength that the plants are perceiving is changing, because here if the shadow is produced by another plant, for example, then they see that there is more far red light coming, and because of that they tend to extend the petals. And it's also measuring in some way the duration of the light or of the illumination, and that's why it's flowering early in long day conditions or late in short day conditions. Then this is why when we started working with plants and in thinking about alternative splicing regulation or gene expression regulation in plants, we actually conceive the idea that light as an environmental cue was able to regulate the alternative splicing and the gene expression in general. But to go a bit further with the perception of light, we know all these events are related to the perception of the direction, the intensity, the wavelength, and the duration. But how do plants see the light? And for this, they have photoreceptors. This can be the blue, let's call them photoreceptors, that are the cryptochromes. The plants also have phototropins and UVV photoreceptor, but mainly what we study is the cryptochromes responses, and there are 2 cryptochromes, 1 and 2. And then on the other side, from the side of the red and far red light, the plants have the phytochromes, and in Arabidopsis there are 5 of them, and these are being regulated by red and far red light. So the activity of the phytochromes is regulated by that. And then at the end of the signaling cascade, you have the integrators, like for example, these high 5 and high H, that are mainly transcription factors that modulate the gene expression of different effectors that are giving a response that would be, for example, photomorphogenesis. So the extension like this of the cotyledons and the biogenesis of the chloroplast, and it's stopping also the growth of the hypocotyl. So these responses are mainly through these photoreceptors that go with the light signal to the integrators, and then these are changing the expression of particular genes. This is true, for example, for germination, where you have the phytochromes. For seedling development, that you also have the phytochromes. So these are probably the more important or the most studied photoreceptors. For the transition to flowering, for shade avoidance, there is this behavior of extending the petioles or the hypocotyl in the seedling. and for tropisms. So all these things are regulated in this way, with signaling pathways that affect the activity of photoreceptors and change the activity of transcription factors or integrators at the end. So we decided to go a bit further, asking whether alternative splicing would be also part of this, not only the signaling pathway, but if it would be important in the responses of the plant to the environment. This is our gene model, our favorite gene. I will be talking about RS31 mainly during the presentation, though we studied other alternative splicing events, because we know for long already that this gene is leading to 3 different alternative splicing isoforms. One of them, the mRNA1, the shorter one that is produced by the elimination, the excision of this whole intron, is the coding one. So this one is actually giving place to splicing regulator. This is coding for an SR protein. And then we have the recognition of these two things, these two lines as introns, and the generation of the mRNA2 that is being degraded actively by nonsense mediated mRNA decay. And we also have the mRNA3, that is the recognition of this as an intron, the use of this 3' splice site here, but this region here that is an intron for the mRNA2 is not eliminated. So you have this whole region retained in the mRNA3, and because of that, this mRNA is not being degraded by NMD, and it's actually, in fact, accumulated in the darkness. So this is the change we see with RS31, that in the light, you don't have much of mRNA3, but in the dark, there is some accumulation of mRNA3. It's actually depending on the duration of the darkness. It can be that is quite equal, that is the frequency or the abundance of mRNA3 and mRNA1. And I will be showing, instead of gels, graphs with the splicing index, and the splicing index is a quantification of this, in which we evaluate the abundance, the relative abundance of the mRNA3 on all the other isoforms. So it's a frequency of mRNA3. And the splicing index is small or low in the light, and it's high in the dark, okay? And with this, yes, the alternative splicing of RS31 is being regulated by light. So this is a good candidate to study how light regulates alternative splicing in plants. And that's the beginning of my work in the last like 20 years already. So the first thing we thought was that because photoreceptors are mainly affecting the light responses in the plants, we said, okay, we will use mutants of these photoreceptors. These mutants should act as blind plants to the light. And we were actually using red light, specific red light, specific blue light, and or white light for these assays. And we were comparing the wild type, Columbia ecotype, with the mutants. For example, this is a mutant of the phytochrome B. This is a double mutant, phytochrome A and B, the main phytochromes in Arabidopsis. And as you can see here, in all the cases, you have the wild type going from white light to darkness. This is the change in the splicing index, and the red and the blue light are also giving a change. So the mutants, all of them are able to see the red light and the blue light, meaning that phytochromes or cryptochromes or whatever photoreceptor are not indeed involved in the perception of light that is regulating alternative splicing. Then we said, okay, but we have a lot of photoreceptors, so maybe there is another one that we didn't test. So we went to the integrators at the end of the signaling cascade, and we use a double mutant high 5, high H with almost no perception of light for many other processes. And as you can see here, in this mutant, we also have an effect of light and dark in the alternative splicing of RS31. So the photoreceptors are not involved in the light-dark signaling that regulates alternative splicing at least for RS31 in Arabidopsis. So we said, okay, let's go back to the eukaryotes and the symbiosis and the beginning of the plants and the chloroplast and everything. The chloroplast is actually another way to perceive the light. Because the chloroplast, for example, we know today that the chloroplast from Arabidopsis has about 100 genes, okay, 100 genes, while the nuclear genome possesses about 4,500 genes that used to be present in the original endosymbiont. So from the nuclear genome, you have to send at least 1,300 proteins that are coded in the nuclear genome, but are working in the chloroplast, while the chloroplast is only generating 100 that are actually working there. So you need some sort of communication between the chloroplast and the nucleus of the plant cell in order to combine and to generate these chloroplasts and making them work. For example, here in orange, you can see the nuclear encoded subunits. This is the photosynthetic electron transport chain in the chloroplast in the thylakoids. And you can see in orange the subunits that are nuclear, and in green, the ones that are encoded in the plastid. So as you can see here, you need equimolecular amounts of subunits that are encoded in both genomes. Then you need a good communication between these two genomes, between these two compartments in the cell in order to make the chloroplast work and actually the plant to live. This communication is actually happening through anterograde signaling, that are signals that are sent from the nucleus to the chloroplast, for example. This is mainly, for example, the RNA polymerase that is transcribing the genes in the chloroplast. That's an anterograde signaling. And a retrograde signaling pathway that is actually signals being sent from the organelle to the nucleus to ask for some particular protein or gene product. So we know now that there are 100 tons of signals like sugars, like reactive oxygen species, and like unknown signals, in fact, that are being sent from the chloroplast to the nucleus in order to get the things that the chloroplast need and to make the cell of the plant to work properly. So with that, we decided to check if the chloroplast was involved in the live regulation of alternative splicing in Arabidopsis. And what we did was to block the electron transport chain of the thylakoids. This is the photosystem 2. And this is the transport to the plastoquinone pool. And by using this EMU, this was called by Bayer-Duron like 40, 50 years ago. And it was a herbicide that is blocking the transport of electrons from the photosystem to the plastoquinone pool. What we saw was that when we added more and more of this drug, this inhibitor, we were blocking not only the electron transport, but also the light signaling pathway. Hence, the chloroplast was indeed involved at some extent. Moreover, we tried another inhibitor of the electron transport pathway, and we block the transport using DBMIV. This inhibitor blocks the transport of electrons after the plastoquinone pool, so the plastoquinone pool is being reduced with the use of DBMIV. With DCMU, it's being oxidized because it's not receiving electrons. And what we saw with DBMIV was that low light was looking as highlight. So this is actually a way to show that the plastoquinone pool and the redox state of the plastoquinone pool, the amount of electrons that are there, is a way to tell the nucleus that the alternative splicing of at least Rs31 must be changed. So if the plastoquinone pool is involved and the chloroplast is involved, then we said, okay, we have the whole seedling. The chloroplasts are only in the leaves. So let's check what's happening in the leaves or in the roots separately. And when we did this, we thought that the leaves will for sure change the alternative splicing in after the treatment with light, and the roots will not affect the alternative splicing because they have no chloroplast. But what we saw was that after 2 hours, no, but after 4 hours of light treatment, then we had this shift also in the splicing index of Rs31, meaning that somehow the roots were able to see the light. However, the roots during this particular assay or this experiment, they were connected to the leaves. Then we decided to do a different experiment, to first dissect the seedlings and then to apply the low light and dark treatment to see if the roots per se were able to perceive the light. What we saw at that moment was that with 4 hours it was not enough, with 6 or with 8 hours it was also not enough. So the roots need to be connected to the photosynthetic tissue in order to see the light, meaning, again, that there is a signal that is being generated in the chloroplast. The signal is traveling throughout the whole plant. And the question, what's the nature of that signal? Well, to make a long story short, we know that photosynthesis is generating sugars, and we thought, okay, then sugars that are actually being generated in light, they can act as a signal per se, as a molecular signal. And what we did was try to use, this is just an osmotic control sorbitol, and to use sucrose, exogenous sucrose, to see if the roots were affected by sugars. And this is what we see. When we did this experiment, when adding sucrose, this is not relevant for the leaves, or not so relevant for the leaves. So this is the splicing index of Rs31 in light, dark after 6 hours. With this is the control, and this is the sucrose treated seedlings. And as you can see, there is still a difference between light and dark. And when we check what was going on in the roots, you can see the effect because these roots were connected to the photosynthetic tissues of light and dark, light and dark. But when we added sucrose, there is no effect. So actually, what we see is that the darkness, it looks like the light condition, meaning that somehow sucrose is mimicking the light effect, mainly on roots. Again, so photosynthesis, you have sugars are traveling through the phloem and reaching the non-photosynthetic tissues or the heterotrophic tissues. We were trying to analyze whether the hexokinases or the SNRKs were involved in the signaling pathway in the roots. And as far as we know, they are not involved. However, we wanted to check also if TOR, the TOR kinase and the signaling pathway of TOR was involved there, as it was reported that TOR kinase is activated in the meristem of the root cells because of the sugar that is coming from the photosynthetic tissues. So we tested this with an RNAi line that has low levels of TOR kinase, and this is what's happening in the leaves of these seedlings treated with light and dark. And what you can see is that in the control, you have a difference in the splicing index of RS31. When you move to the RNAi against TOR kinase, this particular line, though it has low levels of TOR kinase, it is anyway having a difference between light and dark. It's good also to see that the light is increasing the splicing index a bit. So this means that something is doing this TOR kinase in the leaves, but in the roots, the effect is much more dramatic. Here you can see that if you don't have, if you have low levels of TOR kinase, if you don't have TOR kinase in the roots, there is no change in the alternative splicing of RS31 upon light. So TOR kinase is necessary to modulate the alternative splicing of RS31. And if that was true, then we can jump directly to this. By inhibiting the action of TOR kinase, we would have a similar effect. So that's the next thing we did. And this is, again, the control condition with sorbitol and sucrose. Okay, now we are checking whether the effect of sucrose is also going through TOR kinase. When we use an inhibitor of the activity of TOR kinase, here ACD, we see smaller effect of light in the leaves. We see an inhibition of the sucrose effect in the leaves. And what we see in the roots is, again, more dramatic, that you don't have any effect of light in the roots when you inhibit the activity of TOR kinase. And the sucrose is also not adding any effect on top of that. So this means that actually the TOR kinase activity is necessary for changing the alternative splicing of RS31 in roots mainly, not only upon light treatment, but also in response to sugars. And since it was known that the mitochondrial bioenergetics is important for the activation of TOR, we also wanted to check whether this pathway was relevant here in the control of alternative splicing. And we used for that another inhibitor. In this case, is actually dinitrophenol that is acting as an ionophore. So it's affecting the proton gradient in the mitochondria and also in some other membranes. And when using that, here you can see again that in the roots, if you don't have a proton gradient, so if the mitochondria is not working properly, then the roots are not seeing the light. So this pathway, the whole pathway, is active. And to conclude this, we can say that we have a signal, the light signal that is being perceived by the chloroplast. There is a signal that is affecting the alternative splicing in the nucleus of the leaf cells. This is in the photosynthetic tissues. But also there are other signals like sugars that are traveling throughout the plant, and those after glycolysis and after affecting the mitochondria or activating the mitochondria, those affect TOR kinase, and that's in turn affecting alternative splicing in these non-photosynthetic tissues. And though I was mainly talking about RS31, we know that this pathway is relevant not only for 31 in the roots, but also for U2AF65A, RS41, SR34A, SR30, all of these are factors related to splicing regulation. So this seems to be a way for the plants to adjust the gene expression to the light condition. Something interesting was that this is actually mainly from the Alberto Kornblit group, that they wanted to know what was going on in the nucleus. So how this light was being seen in the nucleus. And what they did was to use inhibitors of the RNA polymerase 2 elongation, for example, as here. And what they saw while using these inhibitors was that by inhibiting the elongation in light, they were mimicking the effect of dark. And at the same time, by using a mutant, TF2S, that is also affecting the processivity and the elongation of the RNA polymerase 2, this is also simulating the dark conditions. In general, this means that the elongation of the RNA polymerase 2 in light and dark is different, and because of that, we have this difference in this case, the alternative splicing of RS31, at least. So anyway, I was saying that the chloroplasts are the most important thing in the cell, but you have to know that there are some cases or some reports in which we have the same, but with phytograms. So even though we didn't discover any of these signals from phytograms, there are phytograms that affect alternative splicing and that affect transcription and everything, but it's not the case for RS31 and SR30 and all these splicing factors. But going to the main question is what this is doing. So we know that this change, for example, in RS31 from light to dark, that we have more mRNA3, it's not being seen in the cytoplasm. So it's not giving isoforms that would be translated and that would be producing proteins. Hence, what is this doing? So the mRNA3, for example, and this is the case for SR30 and U2F65 and all these isoforms, are producing transcripts that are nuclearly retained and that would not be translated. So are these proteins doing, are these transcripts doing something, or what's going on? So just to summarize, the mRNA1 is the coding one, the mRNA2 is NMD sensitive, and the mRNA3, we don't know what's going on with that, but we know it's not NMD sensitive, and we know it's quite stable under some conditions. So this I can just jump because I think it's already like an hour. So again, tudo bem, mas ¿para qué sirve eso? Would be the question. So we started to see that we have, when we overexpress the mRNA1, the coding isoform of RS31, we have phenotypes in the plants. You can see that here, that if the plant is exposed to a long darkness period, then it's getting yellow, and actually it can die after a few days. This is the same. Low intensity light generates plants that are pale and that die between 5 to 10 days. And these are smaller. But when we overexpress the genomic construct, that is not only expressing the mRNA1, but also the mRNA3, we can see that this is not having these phenotypes. And it's actually larger, so the plants are bigger in general, they are greener in general, and they can resist some stresses much better than even the wild-type plant. So here you can see another phenotype, flowering. The mRNA1 overexpression is leading to late flowering phenotype, in a short day or in long day, while the overexpression of the RS31 genomic construct is not. You can see the same phenotype in seeds. The mRNA1 overexpression is causing huge, strong germination in non-germinating conditions like far red light. Hence, we started to wonder whether these phenotypes during the whole lifetime of the Arabidopsis plants were related to the mRNA1, but also to the mRNA3. So the mRNA3 is not going out of the nucleus, but is it doing something that is making these plants that are also overexpressing the mRNA1 to resist the phenotype produced by the mRNA1? What we know that the mRNA3 is accumulating in the dark. We know that it's being retained in the nucleus. We know that when we overexpress the mRNA1, there is a shift in the splicing of RS31, the endogenous gene. So if we overexpress this in the wild-type background, there is a shift in the splicing of the endogenous gene. So RS31 is regulating the splicing of its own transcripts. We know that the protein is binding to the transcripts of RS31. So this is one way to know that, but we have also electromobility shift assays and iClip and RIP. So the question is the mRNA3 acting somehow as a long non-coding RNA that is titrating the protein RS31? And we started actually to see that by using, instead of the genomic overexpression and the mRNA1 overexpression, plants overexpressing only the mRNA3 isoform. You see here that there is no flowering phenotype in this plant, at least in this line. But when you put on this mRNA1 overexpression background only the mRNA3, then you can reverse the phenotype of late flowering of this plant. So the mRNA3 might be acting by accumulating in the nucleus and with this part of the sequence and everything as a long non-coding RNA that is titrating the RS31 protein. So our question now is whether there are more RNAs like this, whether this is actually true. We are checking all the other phenotypes and checking the expression of the mRNA3 and the mRNA1 in these lines. We want to also know why this one is being kept in the nucleus while the mRNA2 is going out. We think that's related to the sequence there in this little intron that is remaining. And the next thing would be for us to determine or to evaluate or to actually now it's just a wild guess that coding genes can generate long non-coding RNAs through alternative splicing. Like this is actually blurring somehow the lines between the long non-coding RNA field and the mRNA field, because we have groups that are studying either one or the other. But we think that from genes that are normally known as long non-coding RNA sources, we can generate, the cell can generate mRNAs, and the other way around, from genes that are coding, we can have long non-coding RNAs being generated through alternative splicing. And hence, alternative splicing would be a way not only to generate more proteins to expand the proteome, but also to generate regulatory RNAs. And with that, this is the team. Actually, Lucas was the guy doing the bioinformatic analysis for almost everything I was showing, but he left a few months ago, and Carla was doing the seed experiments that also she finished the master thesis and left. But this is the rest. So these are Lucas left, Carla left, and this is Rocío Toñaca, a researcher in the group, Florencia Rodriguez, a PhD student, Loreana Pulicino, undergrad student, Aldana Kremer, an undergrad student, and Federico Bajay. He's actually studying the interaction between stromules and alternative splicing, and that's me. And with that, just thanks and sorry, because I think it took a bit longer. I was trying to speak not so fast, but then it might be too slow. Slow elongation rate as the polymer.
Thank you very much, Ezequiel. Great talk. And let's
Magda, só avisar os alunos que eles podem, eu imagino que o Petrillo consiga entender as perguntas em português, então os alunos podem fazer perguntas em português, escrever em português ou em inglês, o que vocês quiserem, só não deixem de perguntar, por favor.
Espanhol, italiano, francês.
Perfeito.
Isso, não, e a gente traduz se for o caso, gente. Então, não se preocupem, perguntem.
Magda, você lê aí no chat para eles? Para ele?
Tá, aqui, Gustavo. Ai, peraí. Tchan, tchan.
Primeiro, Gabriel.
Gabriel Márquez, aqui. Períodos curtos de bloqueio da luz durante o dia. Exemplo. Peraí, que eu me perdi aqui, cadê? Socorro, peraí. Aqui. Bloqueo do sol por una nube, por ejemplo, ¿son suficientes para influenciar el proceso de splicing?
Yes. So what we know is that the intensity of the light is affecting the alternative splicing pattern. So if you have low intensity, you will have something similar to dark. If you have high intensity, then you will go to a plateau, reaching high levels of mRNA1 and low levels of mRNA3. But in the middle, you have like a dose response to the intensity of light. However, when you go from light or from more intensity to less intensity or low light intensity or darkness, the effect is quite slow. So if you have, for example, a cloud that is covering the sunlight for a while, I don't think you will have a big effect on alternative splicing. But on the other hand, if you have a cloudy day and then suddenly you have sun in some regions, then that will for sure affect the alternative splicing quickly. In other cases, we were checking long day conditions and short day conditions. In long day, there is, even though there is a shift during the day, it's quite small. But in short day conditions, when you have a long period of darkness, you can see this shift as when we were comparing light versus dark. So just to summarize, the effect is quick when you are going to light, but it's not so quick when you're going from light to darkness.
Ok. Gustavo Ogasaguara. Ezequiel, obrigado pela maravilhosa apresentação e parabéns, felicitaciones por el trabajo. Si los long no-coding RNAs se acumulan en el núcleo, ellos se pasan, digamos, para la F1, para la filial. ¿Qué se sabe de eso?
Nada. but we actually have, in collaboration with Gabriela Auge, she's an expert in this mother and daughter relationships in seeds and between generations. We have the idea that this is happening. Like because of the conditions in the mother plant, you will have more or less, for example, mRNA3, and that will affect the germination of the seed by changing the levels of these let's call them long non-coding RNAs, though we don't really know if this is true. But we have that idea, so I think that would be a wonderful way for the plant, for example, to measure the amount of time that the plant was in darkness or in low light conditions, and to communicate that for the other generation.
Aline Carvalho, profesor, muchas gracias por la clase.
Thanks for the professor thingy.
Como vos demostraste que el alternative splicing en plantas viene aumentando, o sea, el conocimiento, ¿no?, sobre el alternative splicing aumentó mucho a lo largo del tiempo, y ella pregunta cuáles serían los principales avances tecnológicos que permitieron saber eso, digamos, cómo pudo aumentar, que antes creíamos que era 20% y ahora es 60%.
Mainly related to RNA-seq, so transcriptome analysis, mainly related to that. So in plants, though it was being used in animals and it's being used in animals a lot, in plants it was not so common some years ago, and now it's being more common. And that's related not only to the methodology or the capacities, but also because plant genomes are not as well known as animal genomes. So we know a lot about Arabidopsis. We don't know much about other plants. That's one thing. And now, since we have these long read technologies like Oxford Nanopore technology or PacBio, that you might even not need to use a good genome assemble in order to have a transcriptome. Now, I think it will actually boost these technologies will actually boost quite a lot the field in terms of knowledge about new alternative splicing isoforms and events and everything. So we are actually moving to that system to analyze. For example, we think the mRNA3 is one particular sequence, but that's coming from a bioinformatic puzzle that is taking little pieces like reads of 100 or 150 nucleotides and putting them together and assuming that that's coming from the mRNA3. But now with Oxford Nanopore technologies, for example, we can sequence directly the mRNA3 and know if this is the transcription start site and which kind of cap it has and the whole sequence of this thing, even the poly A size. So I think, yeah, it was related to technology for sure, and also to the knowledge about genomes and transcriptomes. But now with the new technology available, I think this will change the field a bit.
Magda, tem as pessoas com a mão levantada agora, eu acho.
Como?
Tem as pessoas com a mão levantada agora, Janaína e o Christian.
Mas só quería acabar aquí que Miguel Romero agradece mucho la clase increíble. Felipe Farinelli también te agradece mucho por esa palestra. Y ahora quien tenha a mão levantada, un momento. Igor, se você quer... Tá.
Christian?
Eu acho que a Janaína levantou
Janaína primeiro. Janaína da Silva Fortuna.
Hi, Ezequiel. Thank you for your presentation. Very nice, interesting subject. My question is, what are the main bioinformatics tools used to study splicing?
Well, thanks for the question. I'm not really good in bioinformatics, so I'm using whatever is easy to use. There is a tool called 3D RNA App Sec tool from the Hatton Institute. This is from the group of John Brown and Craig Simpson. And what they did was actually to generate a package. It's a Shiny app for R. R, I don't know if you use R, but it's this program that you can do statistics and everything. And the ones that you have two kinds of bioinformaticians, the ones that know how to use R and others that use Python and all that stuff. I'm not a bioinformatician at all, but the guy that was working with me, Lucas Servi, he was using Python. So he was doing everything kind of by hand, the scripts and everything by himself. In my case, I'm using this this tool, the 3D RNA-AppSec tool. It's super easy to use, so I fully recommended it. And also for checking the kind of the schemes of the splicing isoforms, there is a web page. I can then send you the link, but it's Boxify. I can write that in the chat. It's Boxify, like that, and you can see the like the cartoons for the splicing isoforms. So I'm using the 3D RNA-AppSec tool to quantify the differences and everything. I'm using Salmon before that for the quantification, and then the tool for the statistical analysis and everything, and then Boxify to see which isoforms are being affected in the different conditions. But I think with almost no knowledge, you can manage to use the tool directly.
Bárbaro. Cristian?
Yeah. Thank you very much. I found it really interesting. And since you were using the 3D RNA-seq app, I wanted to ask you about isoform switching. Have you found any case of isoform switching, maybe on your RS31 gene?
Thanks again for the question also. Yes, I'm not using the isoform switching. They have like this what's the name? Pestaña. Well, tab. That's the name, tab. they have this tab to analyze the isoform switching. I was not using it very often because the treatments I'm using are not actually like being reversed or something. So it's not that I'm using light and another light condition and another light condition. It's just the comparison between light and dark, and that's all. But I was using it at least once for heat treatment that is also regulating alternative splicing in general. And for that, RS31 was not very much affected, but SR30 was being affected. And yeah, after you reverse the heat condition and you go back to 22 degrees or 21 degrees, it's the isoforms P1 and P2 from SR30, the small isoform and the long isoform are changing and shifting like that. So it is a reversible thing, but since my treatments are in general not like that, I'm not using so often this tab.
Okay, interesting. Thank you.
I'm not sure that was the question. Just tell me if.
Yeah, you answered my question. Thank you very much.
Thanks.
Leandro, tem perguntas aí no YouTube?
Tem. I will read for Petrillo. There are two questions on YouTube. First, from Chris Calisto. She's a professor at our department. And
hey.
She's one of the, I think she's one of the developers of the program.
Yeah.
So she said that she's glad to see your talk here in our department. She thanks you. And she's wondering whether you've heard about
She's a really good bioinformatician. She's a really good
yeah, she is.
So you have to ask her.
Yeah, she's wondering whether you heard about light piping. What's your take on that, especially considering your data?
Light piping.
If I'm not mistaken, this like there's some experiments showing that light is kind of like canalized
Yeah.
within the plants?
From the data we have, well, I cannot say much actually. What I can say, because yes, on one side, if piping is what we think, that Christian was referring to, is the idea that these, for example, the hypocotyl of Arabidopsis could act as optical fiber that is allowing light to travel towards the roots. In our experiments, what we saw that was by using sugar or by inhibiting some things related to sugars, we were inhibiting the effects in the roots. So I would consider that mainly, I cannot say much about might be that there is a partial effect of light going through this kind of optical fiber, but mainly the effect we see in the roots for the splicing of RS31 is related to the photosynthesis, like sucrose, for example, not to the light in the leaves directly. We can just remove the roots and add sugar into the media, and they will look as they were in light. So
Okay. There's also another question from Professor Michel Vincent from State University of Campinas. He said that it was a great talk. Sugar coming from photosynthesis modulate alternative splicing, possibly involving mitochondria. Then where does TOR fit in?
Well, TOR is coming after the mitochondria. We don't know how. Actually, the papers that from Shenzhen lab that were the ones discovering this pathway were not very clear in this sense, that we don't know how TOR is being activated after the activation of the mitochondria. But we know that perhaps related to ATP or to any other metabolite that is coming out of the mitochondria after the activation of this organelle, that's activating the TOR kinase pathway. Then how the activity of the TOR kinase pathway is modulating the alternative splicing in the nucleus, that we don't know. If I have to bet or guess, I would say that it's probably related to the translation. So TOR activity is in general changing translation and growth or cellular division, but mainly translation in the cytoplasm. So might be that TOR is activating the translation in general, and you have the translation of particular splicing factors that after TOR activation are going to the nucleus and modulating the splicing in the nucleus. But that we don't know. It's something we wanted to check, but actually working with TOR kinase is not so nice to put it in some sort of way, because it's a very competitive field. It's super expensive. You need antibodies and inhibitors that are super expensive. So we are not doing that much about TOR these days.
Magda, tem uma pergunta lá no chat, eu colei aqui, é lá do YouTube. Uri aceituno, mas eu tenho medo de falar em espanhol e perder alguma coisa.
Don't worry.
Aquí. Gracias, Ezequiel, por la presentación. ¿Hay algún elemento regulador del splicing que pudiera estar permitiendo la unión de algún factor SR o HNRNP que esté regulado por luz?
Yes, it's a very nice question. Yes, we know that, as I was saying, RS31, the protein, it's able to bind the transcripts of RS31. So we think it's binding the mRNA3, but it is for sure binding somewhere in this intron where the mRNA3 is changing the splicing. So we know that RS31, which is a splicing factor, is binding to the alternative splicing event of 31. We think, it's not that this is for sure at all, but we believe, I would say, that 31 after translation is probably binding to this intron that is still present in the mRNA3 and is helping to the recognition of the intron and the elimination. And that's why in light you don't see so much of mRNA3. So yes, we think there is an effect that is related to splicing factors where RS31 might be actually acting, being one of the main factors that is regulating the splicing of its own transcripts. And we know that the binding is possible. We just need to demonstrate whether this binding is affecting the splicing, for example, of the mRNA3 or of the precursor mRNA of 31. That's something we want to know.
Magda, I have a question. May I? Petrillo, you show the effects of TOR, but I don't know if I missed something, but didn't you analyze the effect of the antagonic SNARC? Because they have like this kind of, they have like antagonic functions. So by silencing TOR, you observed an effect. Do you think that the same effect would be observable if you, for example, overexpress SNARC in the same conditions?
Yeah, I was using kin10, kin11 mutants and overexpressors. I was also using exokinase mutants and overexpressors, and I was never seeing any effect. Anyway, yeah, you may have partial effects that I'm neglecting somehow, because it's very hard to know if something is inhibiting a particular process or mechanism if the inhibition is not complete. So I think with TOR, when I saw the gel for the first time and I saw that in the roots was completely abolished, then it was like, okay, this is the thing that is modulating this for sure. It was actually, I was super happy because this was after many years of trying this SNRKs and the exokinase and everything. And at the same time, I was not so happy because it was something that was already known, that TOR was regulating gene expression in the roots. You always want to be more original than that. But I think I was happier anyway than sad, because of the fact that for years I was trying different mutants and overexpression and inhibitors, and I was not seeing any effect on those. It was the same with the photoreceptors. I was trying many times to use mutants and overexpression lines and everything, and I was not seeing any effect until I found that by applying the CMU, that was inhibiting the photosynthetic electron transport, and that was inhibiting somehow the light signaling pathway. This was the same. So yeah, we were using the kin10, kin11 mutants and overexpression lines, and the gene 2, which is the exokinase mutant, the exokinase 1, many things, but we were not able to see any effect. Anyway, might be that there are some partial effects of those.
Alguma outra pergunta? Posso? Tá. Então, eu tenho várias, na verdade, mas já estamos quase 2 da tarde. Petri, in your lines overexpressing isoform 3, how you analyze at what extent the whole transcriptome is affected beyond, the regulation of RS3 particular?
Mhm.
Have you done any RNA-seq or something like that too?
Yes, but we don't have many replicates. That's the thing. So because of a collaborator, we did some Illumina RNA-seq using cDNA, Illumina, and we have one replicate in light and one replicate in dark with these mRNA-3 overexpression lines. And we didn't check yet, but we want to check whether particular events that should be affected by the RS31 protein directly, if those are affected when we overexpress the mRNA-3. So if the mRNA-3 is really titrating the RS31 protein, that might actually affect the function of the protein just by moving it out of the normal targets. So now we have the iClip paper from Maria Kalina and Dorothea Steiger that from RS31 that will tell us whether we know or not particular targets, specific targets from RS31, and then we will check if those targets are affected in the mRNA-3. But that's the main idea. But we didn't do that yet, like a real analysis of the transcriptome, because we don't have enough replicates. It's just one-on-one to do simple comparisons. But we will do it once we have the list of targets of 31.
Okay. And have you done any grafting experiments?
No.
Illuminated, for example, only the aerial part or the roots, and see what happened with that communication?
No, at some point I wanted to do the grafting. At some point, actually, I was talking with a group in Poland that they said they have a technician with small hands that was able to do the grafting experiment and everything. But what I did at some point was to treat one leaf and check what was going on in another leaf, and yeah, the results were not very conclusive. But I don't know. I have to try. We didn't, but in general, what we see is that if you eliminate one part of the seedling or of a plant, for example, because that the leaf experiment I was doing that with adult plants. If you eliminate one or two or three leaves and then you have another that is covered, if you allow the plant to be illuminated for a while, then you can see an effect in the leaf that is covered. So it's not only that the leaf that is photosynthesizing is sending the sugars to the roots, but it's also sending sugars to anywhere where sugars are needed.
Yeah, it is a systemic.
Yeah. Actually, in the same leaf, you have some heterotrophic cells in Arabidopsis, at least.
Mm-hmm. Yeah. Well, I think no more questions. Petri, muito obrigada, muchas gracias.
Please, for you. Gracias para ustedes.
Fue un placer escucharte y bueno, esperamos tenerte otra vez a la brevedad. Okay.
Thanks.
Muchas gracias, Petrillo.
Y estamos ahí, estamos torciendo por la ciencia argentina. Que van a salir de esta.
Fuerza.
Por la selección también, ¿eh? Por la selección.
Bueno, ahí yo me complicás, querido.
Me complicó. Bueno.
Chao,
Muchísimas gracias.